So students, so in the last class what we discussed is Michelson and Stahl has been experimentally proved that DNA as a genetic material. Sorry, what we call as a semi-conservative mode of DNA. So what the Watson and Crick has been theoretically said that there is a possible way where the DNA can replicate itself by what we call it as semi-conservative mode of replication. So semi-conservative mode of replication, in the sense what we're going to see, we will have the half will be newly synthesized and half will be new one. So this is what we call it as the parental and new one will be half will be produced. So that is what we call it as semi-conservative mode of replication. So it has been proved by the Michelson and Stahl by using an experiment called as density gradient centrifugation. So density is created by using cesium chloride. By using the cesium chloride, so the density gradient is created. So this is the both the strands of the DNA, they are of a 15, that is N15 containing. Here one strand is N15, another one strand is N14. So this formation of a hybrid, what we call, this experiment we have already studied. Formation of the hybrid proves that DNA replicates by semi-conservative mode of replication. That means to say what? So two molecules, a DNA will produce two molecules of DNA, where one, that is in the red color, you can see there is a parental and one strand is newly synthesized. So if it goes like that, the red strand completely is going to be reduced and the green strands will increase. So that is actually will be shown in this way, students. For example, you see here, see this is a percentage wise what they have shown here. You can find here, like it was 100% hybrid, it was 100% red, 100% here hybrid. Now the 50%, because 50% hybrid and 50% are newly synthesized, now the hybrid is reducing here 25%, 75%, then again 12%, 18%, 88%. So this way, what we studied in the last class is we come to know DNA replicates by semi-conservative mode of replication. So another one experiment in a Wika, that is Wika Faba. So Faba beans, the same experiment what it is, the one kind of interesting experiment what we can say, Dr. J. Herbert in a Vice Faba, that is Wika Faba, what we call Faba beans. Taylor and his colleagues performed similar experiment on Wika Faba beans using radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosome, and it proved that DNA in the chromosome also replicate in a semi-conservative way. What they have done is the chromatid with the H3 thymidine. So this is now added into radiolabeled thymidine containing medium. So now the radiolabeled thymidine, where one half is the parental, half is newly synthesized, you can see the radiolabeled thymidine. So this is what it proves, semi-conservative mode of DNA replication is right, students. So we see Faba what beans he has done his experiment, Faba beans. So this is what about proving DNA replicates by semi-conservative mode of replication. Now we'll move to the most important and actual concept of how exactly the DNA replication takes place. So DNA replication starts at a point called as origin or ori. You can find here, students, this is a double-stranded DNA. This is a double-stranded DNA where a point where actually the origin, the point of origin, that means to say the DNA synthesis will start. We call it as the origin of replication. How can we identify this origin of replication? Usually this origin of replication will be AT rich region, and what we will find here actually is the double-stranded DNA gets separates into single-stranded. Why do this double-stranded DNA need to be get separated into single-stranded? Because to each of the single strand, you know that in a semi-conservative mode, what we have seen is, so the each of the each of the strand, new strands we are going to synthesize. So they get separate to them, the new strands will be added. One DNA molecule get converts into two DNA molecules. A unit of replication with one origin is called as replicon. So with the one origin unit of replication is there, then we call as replicon. So during replication, two strands unwind and separate. That's what we call it as double stranded strand gets separated and forms into single stranded. By breaking hydrogen bonds in the presence of an enzyme called helicase, students. So helicase is an enzyme where it is responsible for the unwinding the two strands of the DNA, and they are converting them into single stranded. So this unwinding of the DNA molecule at the point forms a Y-shaped. You can find here, this is a Y-shaped. So this Y-shaped structure, what we call it as a replication fork. So what do you will see, students? This is a origin terminus we are having here. Initiation of replication at one origin. This is a one origin where then bidirectional replication at one origin, initiation at the second origin. So this is the bidirectional replication is taking place and initiation or origin is taking place here. Third is bidirectional replication continue until the terminals are reached. So this is what actually the 2 point. This is for starting point where the origin is occurred. So both the way where the process of replication will continues. Then slowly what we're going to see is in both the way, the direction until they'll reach the terminus, what the replication can takes place in a multiple origin site or I having structures. So this is DNA replication starts at a point called as origin. A unit of replication with one origin is called as the replicon, students. So at the origin, what we're going to see, the helicase is an enzyme where it will convert a double-stranded DNA into single-stranded by unwinding it. So this unwinding, what we're going to see, students. So both the separated strands act as a template for the synthesis of new strands. So this is unwinded DNA, for example, if we consider here the unwinding is taking place. So the process is continue, unwinding is taking place here, okay. So unwinding is taking place, a replication fork is growing in this direction. So what we are seeing here is template parental, this is what we call it as template strand. Both of them, they act as a template strand to form a new strands. So in the blue color where you will see here, these are the new strands. So already which are existing in the red color, they are the parental strands. So both the parental strands, they act as what we call it as, what we call them as the template strands. Now, what is clear from the nucleotide formation, students, if you remember, the new polynucleotide chain to form. If you remember this, if you get through this, you will get an idea, students. Polynucleotide chain formation will always takes place in the direction, as I said, if you remember this. So what this is what we call it as, this is a 5 prime 3 prime. So this is 5 prime phosphate already is there. To the 3 prime next phosphate will come and join, and next 3 prime will be available for the binding of next. Here again this 3 prime is available for the binding of next. In this way, this is the 3 prime hydroxyl end, students. You can find here, this is the 3 prime hydroxyl end, where there we're going to see the attachment of phosphate of the next nucleotide. So a nucleotide chain to grow, 3 prime hydroxyl end must be available. This is an idea what I said, explained even during the DNA replication when I was studying about polynucleotide chain. So what do we will see here is DNA replicates in the 5 prime through D3 prime direction only. I hope you are getting it here. So DNA replication starts at the point what we call it as origin. On the origin, an enzyme called as helicase, which will unwind the double-stranded DNA into single-stranded DNA. That's a basic idea what we have. Now, what we will see is after forming the two templates, so to both of these templates, the new strands will be synthesized. How new strands will be synthesized, students? By using complementary base pairing. New strands are synthesized by using complementary base pairing. So we know that what is complementary base pairing. If it is adenine is there will be thymine. If it is guanine here, there will be cytosine. So that way where we're going to see the formation of new complementary strands. Now, so the question here is whether the DNA replication on both the strands will take place in the same way. There is one difference where we can find here, students. To the strand which is moving in this, you see, this is a 3' and this is 5'. To this strand, there will be continuous synthesis possible because here we are going to see 5' end and 3' end will be available continuously. There will be addition of nucleotides are possible. So what is actual requirement? You should get it here, student. This is the most actual requirement. DNA replication can take place only in a one direction, that is from 5' to 3' direction. So that's the reason to the template strand, that is this is the parental strand, it is in the 3' 5' direction. It will to this the complementary 5' 3' direction, it will be synthesized where this is possible in this direction. So 5' to 3' continuous synthesis will take place. Whereas which is in the other parental strand which is already in the 5' to 3', to this actually the 3' to 5' direction. So we know very well that is 5' to 3' direction synthesis is possible, whereas 3' to 5' direction is not possible to synthesize, students. So this will make that the synthesis of the DNA on the template strand, and that is in the 3' to 5' strand and the 5' to 3' strand are entirely different. I hope you are getting here. So the synthesis of DNA on each template strand is different. One which is template strand which is moving in 3' to 5', where it has to have a 5' to 3', that will continuously take place because DNA replication is possible in 5' to 3' direction. Whereas another strand where it is already in the 5' to 3', so here 3' to 5' synthesis is required, but 3' to 5' synthesis is not possible because always a DNA strands to extend what is required, the 3' free end is required, students. So what component is going to provide this, how it is done? So to do this, see, if you see the same diagram, if you hear, students, this is the synthesis of DNA is taking place in a 5' to 3' direction continuously. So the one strand where the continuous DNA synthesis is taking place, it is called as a leading strand, students. You can find here, this is called as a leading strand. The one strand where the DNA synthesis is taking place continuous is called as leading strand. The other strand, if you see, what we call it as lagging strand. So what is lagging strand? Because the 3' to 5' direction DNA synthesis is not possible. What the lagging strand will do is, here it will add a primer. So primer as the 5' end at one side. Okay, this side it has a 5' end and another side 3' end. So the extension will occur 3' to another primer, 3' to another primer. So in this strand, what we're going to see, the discontinuous synthesis of the DNA will occur. The strand where the discontinuous synthesis of DNA is taking place is called as lagging strand, students. So when at the origin the double-stranded DNA gets separates and form into single strand, depending on polarity. So when there are two single strands are formed from the parent strand, the one which is having 3' to 5' will as a complementary synthesis of a new strand in the 5' to 3' direction that will takes place continuously. It is called as this is called as leading strand. The one which is undergoing continuous synthesis is called as leading strand. Whereas another one as a template of a 5' to 3', the new strand has to be synthesized in 3' 5' direction, where it is not possible. Instead, they will add the primer in the middle, then they extend that primer from the 5' to 3' direction only. This is called as conventional 5' 3' direction, where in the reverse way it is taking place, but in a perfect direction. So this is also what happens here is a 5' to 3', again 5' to 3', the direction is becomes same almost. Here also 5' to 3', and here also the 5' to 3'. direction only the DNA will be synthesized, dear students. Now, the strand which is continuously synthesizing is called as leading strand. One strand where discontinuous synthesis is taking place is called as lagging strand. Now, firstly, a small RNA primer is synthesized in the presence of an enzyme called as primase. So, what is a primase, students? See, DNA replication cannot start wherever it wants. It cannot start the process. What is the first and primary thing is required for the DNA synthesis to take place is a 3' end. Okay, as I said, DNA synthesis will occurs in 5' to 3' direction. 5' to 3' direction, the DNA synthesis will occurs. So DNA cannot be started at anywhere else. If the 3' end is available, to that the new nucleotide goes on adding. You're getting my point? DNA cannot be added at any point. So it requires a 3' end. To that the 5' end of the next nucleotide will come and its 3' end will be available. Again the next nucleotide will come join and its 3' nucleotide will be available. In such way the DNA replication will occur, students. So DNA replicates, first the 3' end is required. That 3' end is provided by a primer, that is RNA primer, what we call it, is synthesized by an enzyme called primase. The enzyme called primase will provide 3' end at both the site, either it is in the leading strand or in the lagging strand. So in the leading strand only one primer is enough because it is going to form a 3' end, it goes on get extending. Whereas in the lagging strand, here the one primer will be added, so 3' end will be available, it goes on adding until the next primer comes. Again the next primer it will joins. Then this way the lagging strand has many primers needed to be for the extension, whereas the leading strand only has single primer, students. So in the presence of an enzyme, that is DNA dependent DNA polymerase. So what is that enzyme? The enzyme responsible for the DNA replication is a DNA dependent DNA polymerase. That means to say, by using a template DNA, it can synthesize a new strand of the DNA. Such kind of enzyme is called as DNA dependent DNA polymerase. Students, if you see here it is DNA polymerase enzyme. So DNA polymerase is enzyme where it is capable of adding new complementary nucleotides and extend the strand. So this is what we call it as synthesis of a new polynucleotide chain is done by an enzyme called DNA polymerase. Okay, so now I would like to explain few concepts very clear, students. To understand the DNA replication, what first thing you should know is DNA replication will take place when cell is dividing. That is, the parental DNA has to be divided into 2 molecules of DNA so that it can distribute between the daughter cells. So DNA replication where it takes place during cell division inside the nucleus. It involves the set of proteins. There are various types of proteins which are involved in DNA replication, students. For example, if you have seen here the helicase, where it is helping in unwinding and breaking the double-stranded DNA into single-stranded DNA. As soon as it forms into single-stranded DNA, there will be some proteins called as single-stranded binding protein. They will come and bind at the single-stranded DNA and they will not allow the single-stranded DNA to base pair again and form a double-stranded DNA. So until that part of the DNA is copied, the single-stranded binding proteins will attach there and not allow them to form a double strand. Then the second what we are going to see is the unwinding. When the unwinding is taking place in this direction, so that is the coiled double strand is becoming into single strand. This unwinding will create the super coils here, students. Unwinding will create super coil because ಇಲ್ಲಿ ಬಿಚ್ಚಿದಾಗ ಈ ಕಡೆ ಫಿಟ್ ಆಗಿ ಸುತ್ತಕೊಳ್ತವೆ. ಅದಕ್ಕೆ ನಾವೇನಿರ್ತೀವಿ ಸೂಪರ್ ಕಾಯಿಲ್ಸ್. ಈ ಸೂಪರ್ ಕಾಯಿಲ್ಸ್ನ ರಿಮೂವ್ ಮಾಡೋದಕ್ಕೆ ಮತ್ತೆ ಅಗೇನ್ ದ ನೆಕ್ಸ್ಟ್ ಎಂಜೈಮ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ topoisomerase, what we call the gyrase activity, students. So topoisomerase, what it is going to do is it will cut the DNA and remove the tension which is formed by the supercoiling. So there are various proteins are involved in DNA replication, students. Those proteins and further about the DNA replication, this is just introduction. This is the most important concept that we will discuss in the next class about DNA replication.